Hi friends. So in the last video we saw about the cell theory. What is a cell and what is the cell theory? How was cell discovered and the history about the cells, facts about the cell, etc. And now in this video we'll learn about prokaryotes and eukaryotes. Okay, it's just a very simple topic. Let's move on to the slide. So what is a prokaryote? Prokaryote. The term prokaryote itself denotes the meaning. Pro means before. Okay. And karyotes. The term karyote is also is emerged from the term karyon. Karyon means kernel or nut or nucleus. That means prokaryotes before the nucleus. The major key feature that signifies prokaryotes uh, that detects the prokaryotes is that the prokaryotes lacks a nucleus. They lack a true nucleus. Instead of nucleus, there is a region which is similar to the nucleus and which withholds the genetic material in the bacteria. And this is a very important thing to be known. Okay, whereas eukaryotes contain a nucleus, prokaryotes are devoid of the nucleus. So in this picture, you could see archaebacteria and eubacteria. Eubacteria are the normal bacteria, true bacteria. Okay, archaebacteria. So these methanospirillum, methanobacterium, thermoacidophiles, all these species comes under archaebacteria. Whereas these eubacteria, anabina, gram-positive cocci, gram-negative rods, gliding bacteria, etc. These and all comes under eubacteria. Okay, so let us see what is eubacteria and archaebacteria. So eubacteria are those whose cell wall is made up of peptidoglycan, whereas in archaebacteria the cell wall is made of a different component which is other than the peptidoglycan. Okay, the major differences between the eubacteria and archaebacteria is this is the very important thing. They cannot survive extreme environmental conditions. Who? Eubacteria cannot survive in the extreme environmental conditions, whereas archaebacteria live in the extreme environmental conditions. That is, they can even survive above 100 degrees Celsius, whereas eubacteria cannot survive above 100 degrees Celsius. Okay, this is the major difference between the eubacteria and archaebacteria. But though both of these are eubacteria and archaebacteria come under prokaryotes. Okay, so how is the cell wall? You should know about gram staining. This is very important. This staining technique is used to differentiate between the bacteria. Okay, based on the cell wall components. So gram positive cell wall consists of thick peptidoglycan layer, whereas the gram negative bacteria has a cell wall which is very thin. That is, they contain very thin peptidoglycan layer, and in addition to the peptidoglycan layer, they also contain outer membrane. Okay, now this is a very important technique. You should be aware of this. So what happens is, as you know, bacteria are very transparent and they cannot be seen through the microscope as such. There should be some staining techniques, some color given to the bacteria. So that those color is given through certain techniques. And one of the rapidly used technique is gram staining, which is very important. It employs two stains in the procedure. So in first primary stain, methylene blue, which gives violet color or bluish violet color. Okay, primary stain will be methylene blue. Whereas the secondary stain, which is employed, is the safranin, which gives a pinkish red color. Okay, what happens is the main principle is something like this. When you impart a primary stain, methylene blue, that is a bluish color. Okay, when if it is going to be a gram positive bacteria, it is having a thick peptidoglycan layer. Okay, because of the thick peptidoglycan layer, it can absorb huge amount, enormous amount of the stain, and it can retain even after decolorization. So between the primary staining and secondary staining lies a decolorization step. Okay, decolorization with the alcohol. Okay, when alcohol is when it is decolorized, the thick peptidoglycan layer do not leave the do not leave the stain or dissolve the stain, whereas they retain the blue color. Whereas if it is going to be a gram negative bacteria, because of the thin peptidoglycan layer, they lose the primary stain, and upon decolorization they lose the primary stain, and they take up the secondary stain, namely safranin, and they appear pinkish color. Okay, this is the technique which is employed in the center photo. Picture depicts the gram positive rods, purple color, violet color rods are there, no, that is a gram positive rods bacteria, whereas other pinkish color is called as a gram negative rods. Okay, this is a very important thing to be known. And what are the similarities between archaebacteria and eubacteria? Now we saw the what is the differences between the archaebacteria and eubacteria. What are the differences? The differences, we saw the differences, right? The differences between the archaebacteria as well as eubacteria. Okay, what are those differences? They cannot survive in the extreme environmental conditions. Eubacteria, whereas archaebacteria can survive. This is the major commonest difference. And what are the similarities? Both are prokaryotes. Okay, both have doesn't have nucleus. Okay, there is no membrane bound organelles. Both of them doesn't have membrane bound organelles. They are single celled, that is, they are unicellular, and they are evolved from a common ancestor, and they are microscopic. Okay, now what are the characteristics of prokaryotes? See, this is a bacterial cell. It is having a capsule. Okay, it is having a membrane plasmatica, cytoplasm, ribosomes, plasmids, pili, flagella, etc. The characteristics of prokaryotes is nucleus is absent. So the major characteristic of a prokaryote is the absence of the nucleus. Okay, it has a only nucleoid which withholds the genetic material, namely DNA. Okay, the chloroplast is absent, and the prokaryotes are the oldest cell type. Nucleus is absent, only nucleoid is present, chloroplast is absent, and it is the oldest cell type. See, this is the it lacks the membrane bound organelles like Golgi apparatus, etc. Okay, this is the example of cyanobacteria. Cyanobacteria is something uh, which is a bacteria. All the bacteria, as I said before, are unicellular. Exception is the cyanobacteria, which is multicellular. You can visualize the slide of the cyanobacteria. This is a microscopic observation. The enlarged circular shaped large vesicles are called as heterocysts. They store the uh, food materials inside the heterocyst. Okay. Okay, other features are like they have single circular chromosome, whereas eukaryotes have linear chromosomes. Prokaryotes have single circular chromosome. Okay, and the cell wall is made up of peptidoglycan. Okay, the motility of the bacteria is through the help of the flagella, and they contain the ribosomes, which is of 70S. S is nothing but the speed at which it is a Swedberg unit which is used to measure how fast the molecules move in a centrifuge. Okay, so the 70S ribosome, it has divided into smaller subunit and the larger subunit. The small subunit is 30s, whereas large subunit is 50s, and combined together it is 70s ribosome. Okay, and these prokaryotes range in size from 1 to 10 micrometer. Usually, generally, a common bacteria will range from up to 2 microns, whereas in general, prokaryotes can range up to 10 micrometer. Okay, now let's move on to eukaryotes. What are eukaryotes? Animals, plants, fungi, protists, etc. all belongs to the eukaryotes. What is eu? Eu means true. Karyotes mean kernel or nucleus. It consists of a true nucleus and hence it is called as a eukaryote. Okay, the chloroplast is present. Hmm? And the other membrane bound organelles like Golgi apparatus, ribosomes, endoplasmic reticulum, lysosomes, peroxisomes, all are present in the eukaryotes, which is lagging in the prokaryotes. Okay, they are usually multicellular and the DNA is wrapped around the histones. Okay, and this DNA, when wrapped around histones, it is called as a chromatin. Okay, this chromatin, when it is condensed, it is results in the formation of a chromosome. Okay, chromatin, chromosome, both are same. DNA wrapped around the histone, when it is in the loosely present, it is called chromatin. When this chromatin is condensed further, it results in the formation of chromosome. Okay. So, prokaryotes contain circular chromosome, whereas eukaryotes contain multiple linear chromosomes. Okay. So, the cell wall. What did I say in the prokaryotes? Cell wall of the prokaryotes is made up of peptidoglycan, whereas eukaryotes cell wall is made up of animal cells doesn't contain the cell wall, whereas fungi contains chitin, glucans, mannans, and plant cells contain its cell wall is made up of cellulose, hemicellulose. Okay, and the motility of the cells in eukaryotes is through the cilia and flagella. And what about the ribosomes? Ribosomes are 80s ribosomes. Okay, the smaller subunit is made up of 40s and the larger subunit 60s. On combined together, it is 80s ribosomes, whereas in prokaryotes, it is 70s ribosomes. Okay, there is a theory that suggests that eukaryotes are evolved from the prokaryotes. Okay, now we saw what are the differences between the prokaryotes and eukaryotes. Now let us see about the similarities between the prokaryotes and eukaryotes. So both have DNA. Yes, right. Every cell should have a genetic material. Only then they can replicate. What did I say in cell theory? All the cells emerge from the pre-existing cells. Okay, so definitely both the type of cells should have a genetic material DNA. Okay, both of them have ribosomes. Exactly. Only if the ribosomes are present, protein synthesis can take place. So both should have ribosomes. Both the are contains the cytoplasm, the jelly-like fluid in the center of the cell. Okay, and both the types of cells contain plasma membrane. Okay, now hope I made the point clear. What are the prokaryotes? What are eukaryotes? What are the differences between eubacteria and archaebacteria? What are the similarities between eubacteria and archaebacteria? What are the similarities between prokaryotes and eukaryotes, etc. Hope this video was useful for you. Uh, in the next video, we'll further deal with the types of the membrane bound organelles like Golgi apparatus, mitochondria, nuclear membrane, cell wall. We can learn about or we can recall about these membrane bound organelles in short. Thank you.